Good morning and welcome to the American Society for Microbiology's annual meeting, ASM Microbe, from Boston, Massachusetts. My name is Aliyah Khan and I'm the media manager here at ASM. This press conference will discuss research being presented at ASM Microbe that focuses on the sharing of bacterium related to tooth decay among children and their families. This research provides compelling evidence that children acquire Streptococcus mutans, the bacteria frequently associated with dental caries, from intra and extra-familial sources besides their mother. I would like to introduce Stephanie Momeni, a doctoral candidate in the Department of Biology at the University of Alabama at Birmingham, who will talk about her research. Thank you, Stephanie.
Hello. Yes, thank you so much for having me here today. And as Alina said, I am interested in the bacterium Streptococcus mutans, which is the primary bacterium associated with dental caries or tooth decay. And this bacterium, we're particularly interested in it in severe early childhood caries because I work in the Department of Pediatric Dentistry as well. And what we wanted to look at with this study is, could we track transmission of this bacterium in a large-scale epidemiological study that we've been conducting in a small rural town in Alabama? And what we were able to find with this study, to start with, the literature tells us that we tend to get our strep mutans from our mothers. And this is the prevailing idea, and this is largely because we have a lot of interaction with our mothers when we're very young. It's typically whoever the primary caregiver is in the family. However, there has been research that has shown that we can share strains with other members of our family, and there have been studies that looked at children that share nurseries or are in the same classroom, for instance. They also can share strains, and this is because children very frequently have common sharing behaviors like, here's my ice cream cone, have a lick. And what we've done in our study, like I said, it's a large-scale epidemiological study of strep mutans. And we've followed a group of children and their household family members for the last 8 years, collecting a large repository of about 30,000 strep mutant isolates, of which we've processed about 14,000. We use a typing method known as repetitive extrinsic palindromic PCR, or RET-PCR. I know that's a mouthful. You just really have to remember RET-PCR is the way we typed our bacteria. It is just one method. And what we were able to find with that, we wanted to see if we could track the transmission. And the traditional approach to tracking transmission is to simply ask, is a strain shared with, say, mother, for instance? It doesn't account for whether or not there are other strains that are not shared. And for instance, in our study, we found that children tended to have between 1 and 9 genotypes. And so that simple approach of saying, is there a match, doesn't really capture all of the data. So we did analysis looking at by child, and then by the genotype. So our question was a very simple question. For a given genotype, does the child share that genotype with anyone within the household? And our question of transmission was a little bit more made difficult by the fact that of the 14,000 isolates we've processed so far, we've only discovered 34 RET-PCR genotypes. So that's a lot of commonality of strains. And for a long time, we didn't think we could do anything with the transmission. So again, we go back to our question of, for a given genotype, is it shared with anyone in the household? And our idea is this: if it's shared with someone in the household, we're going to make a simple assumption that they got it from someone in the household, even though we can't rule out the idea that they got it from someone else. But if they don't share the strain with anyone else in the household, we're going to assume they got it from probably child-to-child transmission, like a classmate. And what we were able to find with our data is that roughly about half of the genotypes were shared within the household, whereas the other half of the genotypes were not. Roughly about 157 were shared, 158 were not. And what we also were able to find is that children with, if we look at it by child, we do support the idea that mother-to-child transmission is the primary route. 63% of the children did tend to share at least one genotype with mom. But if you look at it from the genotype perspective, we found that 72% of children had at least one strain not shared with anybody in the household. And if you think about it again, the child sharing behaviors are very common, so this makes sense. And we did observe that children in the younger group tended to have more of these strains. For those strains that were not shared, another interesting find that we had was that roughly about, let's see, a third of the isolates selected, or the bacterial colonies that were selected, were cases where it only occurred one time. And again, this was a longitudinal study. We collected roughly about 70 bacterial isolates over an 8-year period for the children. So to only have it occur one time means it's very, very rare and likely very transient. And why this is important is because in previous studies, they've lumped all this data together. They've not separated it out. And it may mean that roughly a third of the bacteria that we're looking at may have nothing to do with disease. And that may be why we haven't been able to find strains particularly associated with the disease. The next level we wanted to look at is for those genotypes that were shared within the family. And we wanted to look and see how many of them had more than one household family member, and roughly 2/3 had shared with more than one family member, so that supports our idea that they probably got it at home. And then also we wanted to look at mom, because she gets all the blame in the larger picture of things, and to see how many of the genotypes were actually shared with mom. And what we found out is of the 158 cases that were shared within the family, about 85 of those were shared with mom, meaning that there were other sources within the household to be considered. And more interestingly, of those that weren't shared with mom, about half of those were shared with a sibling or a cousin. And that means it adds another layer to the question of like internal horizontal transmission, where it's shared with an older child and they bring it home and share it with their younger siblings. And again, this is probably because of child sharing behaviors. And why this data is really important, why we should care about it. If we can track transmission, it'll make it a lot easier when we identify those strains that are associated with disease to develop targeted treatments and better caries risk assessments. There are a few caveats I'd like to add. Not everyone chose to participate in the study. We did try to get all of the household family members. For instance, fathers participated at a much lower level. Although traditionally fathers tend not to match the child from previous studies, unfortunately they tended to work and they weren't available for collection. And also, this is only one typing method. It will need to be validated with another method just to see if these transmission is truly a characteristic of this population. And also, like I said, it being one method, it needs to be validated, so, okay. Yes?
Hi, I'm Michael Schmidt from This Week in Microbiology, as well as the Medical University of South Carolina. This is a fascinating study, so I'm going to ask you the obvious question. Since Strep mutans is the most common of human infections that we all seem to get, why are we concerned about strains moving amongst individuals, especially children?
Right. Our primary idea for the larger study, why we're conducting the epidemiological study, is that we are curious to see if we can see particular strains that are more virulent and causing much more aggressive cases of dental caries. And we do know that this is possible because it varies from population to population. Even, for instance, among American Indians, there are some populations where caries run rampant and there are others where it's not as severe. So our theory is that there may be more aggressive strains, and we're trying to identify those strains. And initially, we couldn't do that. The data is very dynamic. We couldn't do that because of these extra isolates that were in the mix that probably are transient, and also because of time factors, making sure that we account. It can take up to 18 months after you get a strain to develop dental caries. So there's a lot of factors we're having to take into account that other studies have not done.
So then, if I may speculate, then the long-term goal of your study is to identify those magic strains that could lead to tooth decay.
Right.
And then aggressively follow those children by applying interventions like fluoride treatments, dental varnishes that could then prevent the decay, so the child will then be cavity-free, like
right.
every kid wants to be when they go to the dentist.
Right, and say for instance, it might be one of the things to consider if the child is a stay-at-home child versus a child that's in a daycare. That may be a factor that as clinicians we want to take into account. Also, I'm trying to think of the best way to say this. So the other factor is, we're not trying to say don't kiss your babies. That's an important thing because you want to convey that immunity to your children. But good and bad bacteria do get passed, and it just lets us know that a lot of research efforts are being targeted at mom, and it lets us know, well, that may not be the end-all be-all because they might be getting it from these other children as well.
So in other words, stay tuned.
Yes. We are, it's looking very hopeful. We just had a talk about some genotypes that may be standing out, and particularly in the younger children, because as children get older, their microbiomes become more established. So then other bacteria tend to come in after Strep mutans has set up the environment. And now we're starting to see that if we look at the very younger children, we can really see those that might actually be initiating the disease.
Thank you very much.
Thank you so
Very
much
helpful.
for your question.
Hi, Tanya Lewis from The Scientist. Thank you for your talk. I was curious what you think is sort of the primary route of transmission of the microbes outside of the family, like our kids, sharing toys and like sucking on them, or, and also, so my second question is just, did you look at other strains of other bacterial species and or other populations of children as well?
Okay, and those are both very good questions. So for the first question, let's see. I'm sorry, what was it?
Just what's the route of transmission?
Oh, yes, the mother-to-child transmission, so anywhere where you're using saliva, so in kids are especially young children, they don't have those behavioral norms where we're not supposed to share our drinks, we're not supposed to share our ice cream cone. I always like to use the picture of the child sharing an ice cream cone with his neighbor. Younger children tend to share things, so anywhere where there's saliva being exchanged would be, I think, the primary route. Again, we're not saying don't kiss your baby. And then for your second question, can you refresh?
Just other, did you look at other strains besides the
Yes.
the one
So we did also try to look at Strep sabrinus, which is a very close cousin of Strep mutans. Unfortunately, a characteristic of our population is that they don't have very many Strep sabrinus in this population. We did check that both from the plate methods as well as with quantitative PCR. It just wasn't there in this population. It typically is in others. We did see that Scardovia wigsii was also very frequently common with Strep mutans, and those children tended to have higher caries scores. But that wasn't really part of this current transmission study. This is fairly recent data, so we haven't had a chance to go back. And that's actually one of the things we want to go back is do. This was meant to be a broad analysis to see if it was even worth it since we had so much commonality. And now we want to go back and do a more detailed analysis looking child by child with each family member and the genotypes and was it the predominant genotype, which ones are being shared. But they are, as part of a larger study, looking at microbiome analysis and other factors, whole genome sequencing, things of that nature. Okay, thank you.
Michael Smith, MedPage Today. First question is, but how the study was conducted a little. What I generally cover is HIV.
Okay.
And anytime there's a study in HIV, one of the things that everybody in the study gets is instruction in safe sex and contraception and stuff like that, and STDs. Did you do anything in this study instructing the families and the children in dental hygiene?
Okay. And that is an excellent question. As part of the Uniontown study, and please forgive me, I'm not a dentist, so I wasn't part of the actual pre-screening, so I'll be telling you this secondhand. What they did is they went out to this community center and the schools, and they did preliminary screenings of these children. They did not provide actual dental care because we didn't have the facilities to provide dental care, so just a basic check. And then if they saw that the child had, severe conditions or something that they thought needed treatment, then they referred them to a dentist. One of the reasons why this population was selected is the nearest dentist for these children is 45 minutes away. And when you're in a low socioeconomic group, that's not always feasible. And a lot of times the idea is, well, it's the younger, it's the primary teeth, I'm not going to worry about it till it's in the permanent tooth among these smaller communities. So yes, we did not provide any actual treatments at the site. They had to go to a private dentist. But we do think that nearing the end of the study, because we were providing education and things of that nature, that might have started to change our results near the end of the study.
Yeah, I just wanted to be, I wasn't actually asking about treatment, I was asking about instruction, toothbrushing, flossing.
Oh, yes, yes.
So that was given to
No,
them.
yes,
Okay.
yes, they were given instruction on toothbrushing and flossing.
Okay.
General education type.
And sort of the other question is, there are strains, you say there are strains of S. mutans that are more aggressive than others. Can you, in the research you're doing, track which strains are causing disease in the children, or have you done that?
That is part of what they're looking at right now. Again, we just finished our data collection about 2 years ago, and this is a massive amount of data. And it's not as simple a question as, does it match, does it not match? We were starting to have to go back and account for things, like I said, that time factor of 18 months on average before you get the strain and then you actually start to see the cavities start to occur. So they are looking at trying to determine those strains. And like I said, just before I left, it looked like we had some data that looked very promising as far as about 4 or 5 different strains that seem to be highly associated with dental caries. Okay. And a couple of strains that seem to be protective.
Yeah. Oh, interesting.
Yes.
And a final question is, and this is coming from the recesses of my memory about 20 years ago, it seemed to me there was some interest in either a vaccine or blocking strep mutans in general. Yes. But where are we with that? Is that a viable therapy, or what's going on?
Actually, I believe my mentor, Dr. Noel Childers, was working on a vaccine, and to my knowledge, that's been tabled. I'm not sure of the details for that. I guess it just wasn't as successful as they wanted it to be. So, as far as I know, that's been tabled. It's a very interesting question that you ask, because some groups are looking at trying to remove strep mutans from the mix altogether. And one of my concerns with doing that is, well, if you get rid of strep mutans, are you going to have to worry about periodontal disease or you create another imbalance in the system. So ideally, what I would like to see is that we're able to find some strains that are protective and try to use those strains to create a scaffold that keep away the other bad bacteria. But that would have to be tested to see if those strains would be able to keep the more aggressive strains out, or if they would be outcompeted by the more aggressive strains.
And in any case, that's some ways down the road.
Yeah, it's down the road.
Hi, I'm Laura Beal from Science News. I was wondering if you could talk a little bit more about why it might be important to know this, because I can't see that you're going to be able to stop children from putting each other's toys in their mouth and
right.
Sharing after. So the fact that they share bacteria that way, I don't think, most people would find that, very surprising.
And it's actually very good for their immune system. It's kind of like eating dirt, as children. We don't want to mess up the immune system while trying to stop dental caries. It's interesting to say that basically what we're trying to do is create a better understanding of how we get strains. So like I said, if the child's at a daycare or something like that, the dentist may be more apt to give that child a Mornish treatment because they may be more at risk. It may have to do with the treatment strategies that are going to actually be used by the dentist, or for research studies as well.
Thanks for joining us today, and for all of those who are watching, if you have any questions, please feel free to email us at communications@asmusa.org. Thank you again, Stephanie, for joining us today. That was a great discussion.
Thank you.
Once you're in the BSL-4 space, the only way you can get out is to go through a chemical shower.
It's an unusual room, never seen anything like this. Anybody who has access to this facility first has to go through an R scan.
So the HEPA filters filter the air coming out of the facility, and that will remove bacteria, viruses, anything that might constitute any kind of risk, right?
Remember, this building is basically a second building inside the main building.